So, hi everyone, my name is Jan, and today I'm going to talk about CRISPR, a gene editing technology that can treat genetic diseases. It is estimated that people have between 20 and 25,000 of genes, and just one mutation in a single gene can cause fatal disease. Therefore, a technology that can repair damaged DNA provides a way to treat such disease. CRISPR, which stands for Clustered Regular Interspace Short Palindromic Repeat, is an antivirus immune system of bacteria. It works by capturing and inserting small piece of virus DNA within DNA of bacteria. That creates segment known as CRISPR array. When this bacteria is infected with a virus, CRISPR array will produce an RNA segment which will target virus for destruction by Cas9 protein. CRISPR was first recognized as a gene editing tool in 2012 by scientists Jennifer Doudna, Emmanuel Charpentier, and it was refined by scientist Feng Zhang. It was proven that CRISPR can be effectively used to edit any desired DNA just by using specific RNA templates. So CRISPR has been already used to treat diseases such as sickle cell anemia, and it is a promising tool for treatment of cancer, HIV, and diabetes. So first, sickle cell anemia. It is a disease caused by mutation in a beta-globin gene, one of the genes encoding for hemoglobin. People with this disease have severe attacks of pain, anemia, and stroke. Treatment of sickle cell anemia with CRISPR requires the collecting of red blood cells from a patient, then editing of those cells, editing of beta-globin gene, so that those cells will produce high number of fetal hemoglobin, which has higher capacity for binding of oxygen. And then those cells are reintroduced back into the patient's body. In July 2020, it was announced that the first patient of this treatment had significant improvements. And shortly after, several other patients had improved significantly as well. Next, cancer is a disease that's caused by unregulated multiplication of cells, and T cells that are responsible for their destruction sometimes fail to recognize and kill those cancer cells. So therefore, CRISPR can be used to engineer those T cells so that they will express chimeric antigen receptor, or CAR, which can recognize specific types of cancer. It makes those T cells more effective in killing of cancer cells. Also, CRISPR provides a possibility of using other CAR T cells from other people without being recognized as foreign cells by patient's immune system. Therefore, those off-the-shelf CAR T cells can be produced in great number and stored, and they can be quickly used to treat any person with cancer. HIV is actually an infectious disease, but it embeds its own DNA within DNA of CD4 immune cells, so that it will multiply and then invade and kill other CD4 immune cells. CRISPR can be used to cut off this virus DNA from the CD4 immune cells, and they are currently held a trial to try to lower viral loads of humans with HIV to an undetectable level so that it cannot pass through sexual transmission. And lastly, type 1 diabetes is an autoimmune disease. It causes immune cells to attack beta cells of the pancreas, which are responsible for production of insulin required for metabolism of sugar. People with this disease have to use insulin injections on a daily basis. And CRISPR can be used to edit transplanted beta cells so that after their transplantation to patient's body, they won't be attacked by immune system. And so those people can produce insulin by themselves, and they don't have to use immunosuppressive drugs which cause undesired side effects. In conclusion, CRISPR is a gene editing tool that has potential to eradicate diseases such as sickle cell anemia, cancer, HIV, and diabetes. It is fairly new technology and further research are needed. However, considering its fast development, it's highly possible that this technology will become mainstream, and it can be used to cure many people with different diseases. Yeah, that's my references. Thank you.